And with that, I'd like to introduce our last speaker, Jyotiska Chaudhuri from the Buck Institute is going to talk to us about TRPA1-Nrf2 signaling.
Good evening, everybody. First, I would like to thank the organizing committee for giving me the opportunity to present this work. So what I'm going to talk about today might give us a mechanistic insight in the understanding of diabetes-related complications. So globally speaking, the incidence of diabetes is increasing at an alarming rate. And if we consider pre-diabetes or people susceptible to hyperglycemia, the number can easily reach 1 billion. So what happens in most of the cases is the very early symptoms of diabetic complications is ignored because they are considered as part of life and they eventually become serious problems. So some of the age-related complications in diabetes are highlighted in this slide. And if we just consider neuropathic pain, almost 13 to 16% of the diabetic population eventually develop this condition. And even for pre-diabetic individuals such as impaired fasting glucose or impaired glucose tolerance, the percentage is pretty high as well. So let's take a step back and try to understand what causes diabetes. Diabetes is not just high blood sugar. It's an increase in the amount of glucose, ketones, defective energy metabolism, defective insulin signaling, oxidative stress, and all these eventually gives rise to a class of compounds, actually less detoxification of these class of compounds as well. They are called alpha-dicarbonyls. So two examples of these alpha-dicarbonyls, which are a direct byproduct of glycolysis, are Methylglyoxal and Glyoxal. So these reactive alpha-dicarbonyl compounds are actually 200 to 50,000 times more reactive than glucose, and they are potent glycating agents, and as a result of that, they can irreversibly modify macromolecules such as proteins, DNA, lipids, giving rise to a class of compounds known as advanced glycation end products or AGEs, which has been implicated in a plethora of diseases, including diabetic complications. It has been shown, very interestingly, that a non-diabetic mice lacking Glyoxalase I, which is the enzyme that is required for the conversion of these reactive alpha-dicarbonyls into lactate and glycolate, the mice basically mimics condition of diabetic nephropathy. And recently it has been shown that this is true for diabetic neuropathy as well. So the working model in our study is C. elegans mutant glod 4. It's essentially a C. elegans ortholog for the human Glyoxalase I, and it's a deletion mutant, which is required. The enzyme is required for a conversion of methylglyoxal to lactate. So the first thing that we did in our study is we developed a metabolomic platform to measure these alpha-dicarbonyls. So we developed essentially a highly sensitive platform based on an earlier published study, so that we can accurately measure each and every alpha-dicarbonyl. So on the left side, what you can see is the derivatization protocol. So what we did here is essentially we derivatized each and every alpha-dicarbonyl to a stable orthophenylene diamine derivative. And on the right, what you see is basically individual peaks of each and every orthophenylene diamine derivatives of these alpha-dicarbonyls. So now we have a platform to measure alpha-dicarbonyls accurately in our model system. So with that tool in our hand, we set out to measure endogenous alpha-dicarbonyl methylglyoxal in glod 4 animals. So to our surprise, what we find that glod 4 animals accumulate almost 1,000 times more methylglyoxal compared to N2 animals, and this keeps on going in more and more in an age-dependent fashion. So because of this accumulation of endogenous methylglyoxal, these mutant animals exhibit some very interesting phenotypes. Now, one thing I should mention here, one of the very early symptoms in diabetic complication is a very high sensitivity to touch, temperature, and these kinds of external stimuli. So we wanted to check if we can recapitulate these phenotypes in our mutant. So what we did is we essentially did the simple gentle touch assay. So we used the eyelash, we touch the animal in the head, we reset the touch every time we touch it in the head by touching the animal in the tail, but we only counted the response, reversal response from the anterior touch. And the touch index was calculated by measuring the ratio of the number of positive responses from anterior touch to the number of no response. So what we see here is very interesting. We see that there is an elevation in touch response very early in life, and then they gradually lose sensitivity to this touch. So to follow up this loss of sensitivity in touch, we hypothesized that this could be due to nerve damage. So we checked for nerve damage in glod4 knockdown animals. And we found that this damage is not specific to any particular neuronal area. It's pan-neuronal. So what we characterize the nerve damage, and we see it mostly in the later part of adulthood. We compare this top one is day 10 adult animal, and this is a day 10 glod4 knockdown animal. So we compared the fluorescence in the area of the nerve ring, and we find loss of fluorescence here, which could be due to loss in the cell bodies of the neurons. We find thinning of the neuronal commissures, dorsoventral neuronal commissures, and we also see abnormal branching in some of those commissures. So these were the features that we used to characterize if an animal has a nerve damage or not. So then we kind of quantified this by scoring percentage of animals that harbor any of these damages. What we see here that these damages keep on increasing in an age-dependent fashion in glod4 knockdown animals. And eventually these animals exhibit shorter lifespan compared to N2, which could be due to chronic damage of the nerves pan-neuronally, and they also exhibit a poor handling of glucose. So at this juncture we have a model which is recapitulating many of the conditions of diabetic complications, and this gives us the opportunity to use this animal to answer the long-standing question, how alpha-dicarbonyls mediate disease complications in diabetes. So with that question, big question in our mind, we set out to answer the first question, what kind of transcription factor may be involved in this detoxification mechanism. So we checked a number of transcription factors, but what we found is methylglyoxal is being sensed by SKN-1-Nrf2. So what I am showing here is first, GST4 positive foci. So GST4 is a canonical target of SKN-1. So the number of GST4 positive foci goes significantly high in presence of exogenous methylglyoxal, and then it is significantly reduced in a background of SKN-1 RNAi even in the presence of methylglyoxal. So to further confirm the importance of methylglyoxal in this detoxification, we checked for, we did QRT PCR and looked for expression of another target in addition to GST4. So GCS1 is another target of SKN-1, and both of these are involved in glutathione biosynthesis, which is one of the cofactors in the glyoxalase system. And we find that while in presence of methylglyoxal there is a huge spike in the expression of these genes, this is reversed in SKN-1 RNAi background even in the presence of methylglyoxal. So this suggests that you need methylglyoxal for this detoxification mechanism. Further, we found that SKN-1 has a protective effect against these phenotypes. So in the background of SKN-1, the animals are more sensitized in glod4 knockdown in glod4 knockdown animals. So you have a higher sensitivity to touch in SKN-1 mutant background, and the lifespan is further shortened in this background as well. And then we wanted to check if we can rescue this phenotype tissue specifically. So animals were used which specifically expresses SKN-1 either in the intestine or in the neurons, and we found that intestinal SKN-1 is required for this rescue in both for the touch sensitivity phenotype and for lifespan. So it seems like we have a transcription factor which seems to respond to MGO toxicity downstream, and then we asked how is methylglyoxal sensed upstream, and we hypothesized that this could be due to membrane-bound ion channels, because very recently there was a paper in mice which shows that sodium channels are involved in neuropathic pain. So with that, we set out to check for membrane-bound ion channels, and we particularly looked into TRP channels. The reason is our mutant exhibit mechanosensory phenotype, mostly touch sensory phenotype, and TRP channels are conserved thermo and mechanosensory channels, which are conserved across the phyla. And very recently the structure of TRPA1 was actually found, and if you see the structure, you can see that there is a hinge-like structure which modulates the movement of cation across the membrane. So these are non-selective ion channels, and the majority of the TRPA channel has a lot of structural conservation. And what we find, so we basically checked for which of these TRP channels could be involved. So we did, first we checked for lifespan, and we checked lifespan in glod4 knockdown animals in the background of all the available TRP channel mutants. And we found that in the background of TRPA1, the lifespan is further shortened. So this indicates that there is a requirement of TRPA1 in this detoxification mechanism. And because TRPA1 is because we know that there is an importance of intestinal SKN-1 in this detoxification, and most TRP channels work through active ion signaling. We used a transgenic animal carrying a GFP tagged calcium reporter to check for potential calcium flux in the intestine. So what we find is that methylglyoxal, under methylglyoxal evoke, there is a robust response in this transgenic animal for calcium flux, and this response can be reversed by a TRPA1 channel mutant which is specifically impermeable to calcium flux. So based on these two slides, we can summarize that number one, there is a requirement of TRPA1 in this detoxification mechanism, and number two, there is a requirement of an active calcium flux to transduce this signal. So the next question we asked is whether TRPA1 channel senses methylglyoxal directly, and whether this response is conserved across the phyla. So what we did is we expressed TRPA1 channel in human embryonic kidney cells which do not express TRP channel natively, and we checked for calcium flux. So what we find that there is a robust increase in calcium flux in a dose-dependent manner in both rat TRPA1 and both by worm TRPA1. So that means that TRPA channel is directly sensing methylglyoxal, and this is conserved across the phyla. And then the next question we asked is if this response is specific to TRPA1. So we used another channel as a control, which is a TRPM8. This channel is specifically responsible for sensing menthol. While it was able to sense menthol, it failed to respond to methylglyoxal. So that rules out the possibility of at least TRPM8 in this detoxification mechanism. So then we asked the question, what about any other agonist? So because TRPA1 has a lot of agonists, and one of the very classical agonists is allyl isothiocyanate, which is an active ingredient of wasabi. Now, in an earlier paper, it has been shown that allyl isothiocyanate failed to evoke any response in worm TRPA1. The reason is it requires 3 cysteine residues which are lacking in worm TRPA1. So we wanted to check if that is true for methylglyoxal as well, and how does it stand. And we know that from my previous slides that methylglyoxal can evoke worm TRPA1 and it can evoke rat TRPA1, while AITC can only evoke here in rat TRPA1. So this suggests that the mechanism by which methylglyoxal is triggering response in TRPA1 channel is a distinct mechanism from a classical TRPA1 agonist like AITC. We are actually working on this to understand what could be the potential mechanistic pathway of TRPA1 activation by methylglyoxal. So now that we have a upstream receptor or a channel that receives this signal, and we know that there is a requirement of downstream SKN-1-Nrf2 in this detoxification mechanism, we ask the obvious question of epistasis, whether these two elements are talking with each other. So to do that experiment, what we did is we checked for GST4 positive foci, which is a SKN-1 target in the background of TRPA1 RNAi. And we see that the number of foci is significantly reduced in the background of TRPA1 knockdown. And this was true for the expression like before for GST4 and GCS1 as well. In the background of TRPA1 knockdown, the expression of both these genes, which are SKN-1 targets, was significantly reduced. So if all this is true, then definitely our metabolomic studies now will prove that if this pathway is working in a detoxification mechanism, we are supposed to get a higher level of these alpha-dicarbonyls in the background of these genes. So to check if this is true, we used our platform to check for alpha-dicarbonyls. And here what you see here, two backgrounds, N2 and then glod 4. And in the background of N2 and glod 4, we checked for the levels of the alpha-dicarbonyls in both TRPA1 and SKN-1. Where you see here there is a 530-fold increase in the levels of methylglyoxal in N2 animals. Glod 4 already had a huge amount of methylglyoxal to start with, but there is still a 3.5-fold increase in the background of TRPA1 and SKN-1 RNAi. So this particular spike suggests that besides glod 4, there could be other glyoxalases that are also playing a critical role in this detoxification mechanism. So we know what glyoxalases are playing in this mechanism, but for the limitation of time, I will not tell that story today. So this gave us a novel signaling axis which seems to be working for the detoxification of alpha-dicarbonyls, and this could easily be a potential pharmaceutical lead. So with that goal in mind, we set out to do a drug screen. We did a NPL 640. These are natural product library. This is a natural product library of 640 compounds, a screen to look for any hit that could ameliorate any of these phenotypes exhibited by glod 4 animals. So we only screened for phenotypes that was able to ameliorate all of the screened for drugs that could ameliorate all of the phenotypes exhibited by glod 4, and we got 5 or 6 hits. Out of that, PKL1 is one of those hits, and it was able to ameliorate all, as I mentioned, all the 3 phenotypes. So it was able to ameliorate the touch sensitivity early in life, the loss of sensitivity later in life. There was significant amelioration in the nerve damage phenotype, and it also extended lifespan only in glod 4 animals. So 2 examples what I have is in the legend here. One is alpha-lipoic acid and another is UMP, which is uridine monophosphate. So uridine monophosphate was one of the controls from our library, and it is interesting because uridine monophosphate came out of the touch screen, but it was not able to ameliorate any of these phenotypes. So we kept it as a control, and alpha-lipoic acid, on the other hand, is a supplement used for diabetic patients in many other countries, and that was also able to rescue the lifespan of glod 4 animals. So then we wanted to check if this amelioration in health span and lifespan of glod 4 animals is due to reduction in alpha-dicarbonyl. So we did MGOPD measurement in the treatment by treating the animals with PKL1. LA and UMP also act as a control here. And we saw significant reduction of alpha-DC in the presence of PKL1. So then we wanted to check if this is conserved across the phyla, and if this drug can ameliorate neurotoxic effects of these alpha-dicarbonyls in rats. So we used an immortalized rat DRG neuronal cell line, and we measured the health of this cell line by measuring neurite length and soma area. There was a significant improvement in neurite length on PKL1 treatment, and also for soma area. But because this concentration, we started with a higher concentration to check for this effect, but we did follow this up with an EC50 study to look for the efficacy of the drug at a lower concentration, and there was amelioration at a lower concentration, and it's in a dose-dependent manner. So to conclude my talk, I would like to summarize the major points. So we have a model system now to look for alpha-DC mediated toxic effects. It's glod 4 mutant, which is carrying a deletion mutation in the Glyoxalase I gene. We have found a direct sensor for methylglyoxal TRPA1, which is conserved across the phyla for sensing methylglyoxal. We see that conserved glyoxalases downstream play a critical role in detoxification of these alpha-dicarbonyls, and there is a direct crosstalk between TRPA1 and Nrf2 in this detoxification mechanism. And last but not the least, we found a novel drug that could ameliorate alpha-DC mediated pathologies by lowering levels of alpha-dicarbonyls. So this is our model. I spared you from explaining all the intricate details of the model because of limitation of time. So we found that TRPA1 sensing alpha-DC opens up. There is a robust increase in calcium flux, which is received by ANC43, which is a CAM kinase 2. This signal is then transduced through a battery of p38 MAP kinases, which then is received by SKN-1-Nrf2, and that transcribes a bunch of glyoxalases, glod 4, which is the glutathione-mediated glyoxalase, which is working in complementation with another glutathione-independent glyoxalase, which is DJ1. I haven't shown you the data of these glyoxalases. And then another glutathione-independent glyoxalase, which is DJR1.2. With that, I would like to end my talk, but before that, I would like to acknowledge all these people for their support and help, and for NIH for my funding. Thank you very much for your attention.
Interesting study. So my only question was, I was wondering if you screened this natural product library on the HEK cells expressing the TRPA1 receptor or channel, or did you just bypass that, or?
Yeah, we did not check the cell for the metabolites.
So this doesn't maybe I'm confused. So this natural product that you found,
yeah,
that doesn't interact directly with the TRPA1 receptor.
Oh, yeah, it does. I haven't shown you the data. So the agonist that we find, or the drug that we find, is also a TRPA1 channel agonist. Right now, as I speak, we are checking if that is true in cell culture as well.
Okay.
Yeah. Thank you.
Hi. I was in the last slide. Did you indicate that SKN-1 is driving expression of glod1 in response to the Methylglyoxal?
Yes.
Okay.
Yes.
So it acts at the level of
Yes.
transcription.
Yes.
And what is the biological significance of GST4, GFP foci?
So the reason, we just used as a reporter for SKN-1 activation.
Okay. But there's
it is involved in glutathione biosynthetic pathway.
Right, but there's a total increase in GST4, GFP also. There's an overall increase of GST4, GFP.
Yes.
Not just these foci.
No.
Okay, let's thank all of our speakers.
Thank you.